This lecture is going to review some information about enzymes that will be helpful to you when you write your lab report. Most enzymes are protein molecules. There are a few that are RNA molecules, but the ones we'll be concentrating on in Biology 1 will be protein molecules. Because they are protein molecules, enzymes are held together by hydrogen bonds and covalent bonds. Enzymes are referred to as catalysts. Catalysts speed a reaction without being used up in the process. The enzymes that an organism has depends upon its DNA. If you recall the central dogma of biology, DNA is the template for RNA, and RNA is the template for protein. Therefore, any enzymes an organism produces will be coded for by the genes that an organism has. In other words, it's DNA. Enzyme names usually end in ase. The enzyme that you used in lab is called catechol oxidase. An enzyme that would break down a lipid, for example, would be called a lipase. Enzymes that break down sucrose would be called sucrase. Now, this is not a hard and fast rule. There are some enzymes that don't end in ase, but generally, if you see that suffix ase, you are probably dealing with an enzyme. The molecules upon which enzymes act are called substrates. Metabolic pathways are a series of linked reactions. Later this semester you'll be learning about several reactions that we see in the cell, including photosynthesis and respiration. And you'll be looking at these pathways. Some of them are linear, some of them are circular. But as you can see in this pathway, there are a series of reactions that are linked to one another. The product of a previous reaction becomes the reactant of the next reaction. So, for example, the letters A through G indicate substrates, whereas the letters E1 through E6 represent the enzymes. So E1 is going to have the substrate of A, and the product of that reaction is going to be B. The substrate for E2 then is going to be B, and the product is going to be C, and so forth. Molecules frequently do not react with each other unless they are activated. The energy of activation is the amount of energy needed to cause molecules to react with one another. Now, sometimes heat is enough to do this. Heat lowers energy of activation because it speeds molecular motion. As molecules move faster, they're more likely to bump into one another, and a reaction can happen. This is a bit of a problem for a cell, however, because when you turn up the temperature, you can actually destroy the cell, and you can also destroy the enzyme. So enzymes are used by cells in order to lower the amount of energy required for the reaction to take place. Enzymes act as a stage of sorts. They bring substrates into contact with one another, and they sometimes even participate in the reaction. After the reaction takes place, the products are going to be released, and then the enzyme can be reused. Here we can see a slide showing a graph showing the energy of activation. And you can see that without the enzyme, a great deal of energy is necessary in order to convert reactant to product. On the other hand, when an enzyme is present, far less energy is needed to convert reactant to product. A lower hurdle is easier or faster to get over, so a lower energy of activation makes a reaction easier or faster. Enzymes lower the energy of activation. So here you see an image of an enzyme. This is an artist's rendering, but enzymes have shape. So protein molecules are going to have that three-dimensional shape. You can see that they're very much like locks, and a key will fit into them. The key is the substrate. So you see this blue molecule, and it has a particular shape. And you look at the substrate, and you can see that the substrate has a corresponding shape. The enzyme is going to be attracted to the substrate. They actually just bump into each other, but the fit is just about right. And so when the substrate fits into the enzyme, a reaction is going to occur, and the products are going to be released. Now, enzymes can be involved in synthesis reactions where they bring together A plus B to make C, or they can be involved in degradation reactions where C is broken down to A plus B. Enzymes have an active site that accommodates the substrate. It's like a lock and key, specific to one substrate. So enzymes are going to be specific for what will fit into the active site. When an enzyme binds to a substrate, it's not a 100% great fit. It's sort of like having a pair of jeans that are a little bit too tight. So when the substrate fits into the enzyme, there's a slight change in shape to accommodate the substrate. This is going to put stresses on the bonds and is going to facilitate product formation. The active site, after this change, returns to its original shape after the products are released. Enzymes, remember, are not used up by the reaction, so they can go on again and be used. Now, sometimes enzymes need activators. For example, cofactors. Cofactors are inorganic molecules, this means they don't have any carbon associated with them, that help with the chemical fit of the substrate to the active site. They're going to be permanently bound to the enzyme, usually in the active site, and very often they are metal ions. If you take a look at your vitamin bottle, for example, you're going to see there are a lot of metal ions that you take every day in your daily vitamins. This is going to help your enzymes work properly because the substrates will fit in appropriately into the active sites. There are some examples that are given here, and I'm sure you notice on your vitamin bottle that you take zinc or you take iron. Iron is Fe. Copper is another one, Cu. Potassium, K, and Mg is magnesium. And some of the enzymes, you can see most of them here end with ACE again. So the enzymes or proteins that are going to be helped with these cofactors are listed. Copper, for example, is needed by us for an enzyme known as cytochrome oxidase. Copper is a cofactor for the enzyme that you used in lab called catechol oxidase. Without copper, the enzyme is not going to function properly. So cofactors are going to be part of the active site, and they are going to be permanently bound. They help with the chemical fit. Then there are also coenzymes. These function differently. They're organic molecules, so they're going to contain carbon, and they participate in the catalyzed reaction by transferring subatomic particles, such as electrons or protons. They can transfer molecules, or they can transfer functional groups. They are not permanently bound to the enzyme, and you'll be learning about some of them later this semester, such as NAD, nicotinamide adenine dinucleotide, or FAD, flavine adenine dinucleotide. Coenzyme A is another one that you're going to learn about this semester. And you can see that NAD is going to participate in the transfer of electrons, whereas biotin is going to participate in the transfer of carbon dioxide, or CO2. Enzyme inhibition occurs when an active enzyme is prevented from combining with its substrate. An example is cyanide. Cyanide is a poison because it binds to iron in cytochrome oxidase A3. Cytochrome oxidase A3 is found in our mitochondria, and it's involved in cellular respiration. If cyanide binds to this enzyme cytochrome oxidase A3, oxygen is prohibited from binding and cellular respiration ceases. In other words, the cells suffocate. There are two types of enzyme inhibition. One is called competitive. The other one is called non-competitive or allosteric inhibition. Competitive inhibitors are molecules with a similar shape to the substrate, and they bind to the active site. So if the key is very similar, the key is going to fit into the lock, but it's not going to turn it. This has probably happened to you when you went out to the parking lot and you saw a car that was a lot like yours. You thought it was yours, you put the key in the lock, but the key wouldn't open the car. Okay. This is an example of a competitive inhibitor. Now, competitive inhibitors compete with the substrate, and hence the name competitive inhibition. So if you add additional substrate, you are going to get product. It's a competition. So if there's more substrate, you'll get product. If there's more inhibitor, you won't get product. So if there's more competitive inhibitor than substrate, the reaction is going to slow. If there's enough of the competitive inhibitor, the reaction will stop. There's an example of an antibiotic that you might have taken at one point for a urinary tract infection. It's called Bactrim. This antibiotic is going to affect bacteria. it competes with a substrate that bacteria need to make DNA. That substrate is called PABA, and the competition prevents the organism from making DNA. We don't use this enzyme that the bacteria have, and so it doesn't affect us at all. If you've ever taken this antibiotic, you know you have to take it several times a day to keep the competition going. If you forget to take this antibiotic, the organism will just use the natural substrate and it will create DNA and it won't die. It will continue to multiply and grow. Here's an example of competitive inhibition. You can see here where there's an inhibitor molecule. Now this is the normal substrate shape right here, and this is the shape of the inhibitor. The inhibitor fits into the active site, and it is going to prohibit the substrate molecule from fitting in there. You can see the shape is very much the same. Now if there is more inhibitor than there is substrate, then the inhibitor is going to win and the product won't be made. But if there is more substrate than inhibitor, the substrate will still fit into here and product will be made. Now a different type of inhibition is called non-competitive inhibition. We also refer to this as allosteric inhibition because the inhibitor binds to a site other than the active site. The word allosteric means site other than, so we call this the allosteric site. The inhibitor either blocks the active site or it changes the shape of the enzyme's active site so that the substrate cannot bind. So here's an example of a non-competitive inhibitor. It's binding to that allosteric site here, and it is blocking. So the shape of this inhibitor actually blocks the active site, so the substrate cannot bind to the active site. There's a second type of non-competitive inhibition. This one doesn't involve the allosteric site. Here we have the inhibitor actually binding to the active site of the enzyme itself and blocking the substrate from getting in. It's sort of like putting a boulder into the active site, so the substrate just can't fit into here. Non-competitive inhibitors can also bind to the active site and block the substrate from binding. There is an example in lab, which is phenylthiourea. Phenylthiourea binds to copper. Remember, copper is that cofactor in the active site of catechol oxidase. Well, when phenylthiourea binds to the copper, it's like a boulder blocking the entrance of the substrate catechol into the active site of catechol oxidase. Now, it doesn't matter how much substrate you add in a situation like this, simply because the copper is bound tightly to this inhibitor and it's going to continually block the substrate. In this case, we have an allosteric inhibitor who actually changes the shape of the enzyme. So you see here the substrate molecule. The substrate molecule would fit into the active site. There's also an allosteric site present in this example, and there is a molecule, the allosteric inhibitor, that will fit into the allosteric site. An allosteric inhibitor binds to the allosteric site. When it does this, it changes the shape of the molecule so that the active site no longer has the original shape, and as a result, the substrate cannot bind. Another type of non-competitive inhibition is called feedback inhibition, and this is very important in living organisms, especially when a pathway is involved. Here you can see the enzyme. There's an active site. The substrate is going to fit into the active site, and there's also an allosteric site present on this enzyme. In this case, we see that there are several different products, several different enzymes, and the end product, as long as it's being taken away, used up by the organism, the product doesn't accumulate, so the reaction just keeps on going. But sometimes the product might not be needed anymore, and the product is going to accumulate. When this happens, the product of that reaction will fit into the allosteric site. So here you can see the product fitting into the allosteric site, and look what happens to the shape of the active site. It changes. The product binds to the allosteric site, changes the shape of the active site, and when the shape is changed, we call this denaturation. And when we lose the shape, we lose the function. So here you see now that the product is binding to that first enzyme in the reaction. The substrate no longer can fit into here, and as long as this happens, then no more substrate will be converted to product. Now this isn't a permanent bond here, so if the product is going to be used up again, this product is going to release from this enzyme. The active site shape is going to go back to the way it was, and the substrate will then fit back into it. We can also look at the rate of reactions when we increase the amount of enzyme that's present. When substrate is held constant, adding additional enzyme will speed the rate of the reaction. That makes sense. You press on the gas pedal more, your car is going to go faster. Okay? The availability of substrate will be the rate limiting factor. So once you run out of substrate, no more product is going to be made. So you can add enzyme, and as long as there's substrate, adding more enzyme is going to increase the rate of the reaction. But when substrate runs out, then you won't have the reaction occurring anymore. Now this is a little bit different when we increase the amount of substrate. You can see, looking at this graph here, that increasing the amount of enzyme increases the rate of the reaction. But take a look at this graph. Okay? When enzyme is held constant and substrate is increased, you can see that we do have an increase in the rate of the reaction, but then eventually you reach a point where the reaction plateaus. What's happening here? Well, we call this saturation. The available enzyme molecules are saturated with substrate. Every single enzyme molecule in there has a substrate in it, and there's so much substrate in that reaction that it's almost like a traffic jam. Think of a tunnel and the traffic jam before a tunnel. If you think of the cars as being the substrate and the tunnel as being the enzyme, eventually all of those cars, or all of those substrate molecules, are going to get into the tunnel. But they're going to be slowed from doing this because there are so many of them trying to get into the tunnel at the same time. So the reaction rate, when you increase substrate, will go faster at first, but then eventually it will slow when the point of saturation is reached. In lab, you looked at a number of environmental factors that affected enzyme action, so let's take a look at these. Because enzymes are protein molecules, anything that can denature proteins can affect enzyme function. All enzymes are going to have optimum temperatures at which they function, and also optimum pHs. The enzymes in your body, for example, are going to work best at 37 degrees Celsius. And the enzymes in your blood are going to work best at a pH of 7.2 to 7.4, because that's the pH of your blood. The enzymes in your stomach are going to work at a much more acidic pH, because that's the pH that's in your stomach. Exceeding these ranges are going to cause the protein to fall apart. In other words, the hydrogen bonds will break and the enzyme will denature. When an enzyme is denatured, the active site no longer has the shape that it originally had, and therefore the substrate can't fit into it, and it will no longer catalyze the reaction. If the optimum temperature is exceeded, hydrogen bonds are going to break. Here you can see part of a protein molecule, and these little red dots indicate the hydrogen bonds. When the temperature gets too high, just like with water, hydrogen bonds are going to break. Now cold temperature, on the other hand, cause hydrogen bonds to get stiff, so they push away from one another. Remember when we studied about water? Water expands when it freezes, so those hydrogen bonds push away from one another. So if we're dealing with a protein molecule and those hydrogen bonds now are expanding, this is going to expand or change the shape of the active site, and therefore the substrate is not going to fit into it anymore. So using cold temperatures and high temperatures, both will affect the hydrogen bonds of enzyme molecules. pH can also change. Altering the pH can also change the shape of the protein molecule. If you add an acid, for example, to the solution, this denatures the protein. So oftentimes when you cut up an apple, you put some fruit fresh or you use some lemon juice on it to prevent it from browning. Okay? Acids are defined as hydrogen ion donors. For example, hydrochloric acid will donate hydrogen ions. Now think about this. If you have that nice hydrogen bonding going on with electrostatic interactions between negative areas and positive areas of the molecule, and now you throw in a bunch of positive ions, that's going to disrupt that very delicate balance. Additional hydrogen ions will disrupt the hydrogen bonds holding together the molecule. This is going to cause a change in the active site, and therefore the substrate molecule will not fit into the active site anymore. Sodium chloride or salt pretty much does the same thing. Remember the solvency property of water? You know that if you put sodium chloride in water that it's going to dissociate into sodium ions and chloride ions. The presence of these ions will disrupt the hydrogen bonds, just like adding excess hydrogen ions to it, and this will denature the protein molecule.